Okay, so let's dive in. We're starting this deep dive looking at, well, the blueprint of life, really.
Hmm.
And a fascinating place to begin is the story of Dolly the sheep.
Ah, Dolly. Back in 96, she
Uh,
was, well, she was a huge scientific success, wasn't she? A real game changer.
totally. Because she showed that scientists could just completely sidestep the usual way reproduction works.
Exactly. She was, a clone, genetically identical to the sheep that donated a single udder cell.
No father involved.
Yeah.
Which is kind of the opposite of how most complex life passes on its genes.
Right. And what's really wild when you think about it
Yeah.
is that this cloning, taking a body cell and making a whole new organism, it was only possible because of like a century of research.
Right, understanding the basic rules first.
Exactly. Scientists had figured out the fundamental ways cells normally divide and handle DNA. Knowing those rules let them, well, break them.
So that's our mission today, for you listening. This is your shortcut to getting your head around that rule book.
Yeah, we're diving deep into the cells, the chromosomes, how mitosis and meiosis work.
And how all this plays out in the life cycles of different organisms that scientists actually study, from cell basics to diversity.
Let's start right at the bottom then. The cell. Life happens inside this little membrane-bound bag, basically.
So what's inside, in that cytoplasm?
Well, it's mostly water, but you've got all the essential molecules, carbohydrates like starch for energy,
Uh-huh.
lipids, making up membranes, giving structure, and then proteins, the real workhorses doing everything, especially enzymes.
Catalyzing reactions.
Yeah.
And the instruction manual, the really key part.
Always the nucleic acids, DNA and RNA. That's the genetic blueprint.
Okay. Now, when we look at cells, there are sort of two main types, aren't there?
Broadly, yes. First, you have the prokaryotes. Think bacteria, archaea.
Smaller, simpler.
Much simpler.
Yeah.
They don't have complicated internal membranes, no specialized compartments or organelles, really. And their DNA isn't tucked away in a nucleus. It's just there.
And the other type, eukaryotes.
Right. That's us, plants, fungi. Usually about 10 times bigger. And they do have all that complex internal stuff.
Like the endoplasmic reticulum, Golgi.
Yeah. And organelles for energy, like mitochondria or chloroplasts in plants. But the defining feature, the big one,
The nucleus.
the nucleus. A membrane wrapping around the hereditary material, the DNA, keeping it organized and safe.
And inside that nucleus, the DNA isn't just a long string, is it? It's packaged.
Very carefully packaged into structures called chromosomes. A chromosome is basically one super long DNA double helix wrapped around proteins.
Okay, and the number of these chromosome sets matters. That's ploidy, right?
Exactly, ploidy state. Most of your body cells, what we call somatic cells, are diploid. We use the notation $2.
2 copies of each chromosome, the full set instructions.
That's the idea. One set from your mother, one from your father, inherited through the egg and sperm.
Which brings us to the sex cells, the gametes. They're different.
They have to be. They are haploid, just no allers. They carry only one copy of each chromosome, half the manual if you like.
And that's crucial for sexual reproduction.
Absolutely essential. These haploid gametes come from specialized diploid cells in the germline.
So during fertilization, a haploid sperm fuses with a haploid egg.
And boom, you're back to the diploid state, the two-lay zygote, the first cell of the new individual, with the complete genetic instructions restored.
Okay, makes sense. So we know what needs to be copied, the chromosomes. How does the cell actually do the copying and dividing? There's a cycle, right?
A very precise one. The eukaryotic cell cycle, it goes in stages. G$1, which is growth. And then the crucial Zeller phase. S for synthesis.
That's the DNA copying part.
That's the DNA copying part. Every chromosome gets duplicated. Then comes G$2, more growth and preparation.
And finally,
Finally, an Aller phase. M for mitosis, which is the nuclear division, followed by cytokinesis, the cell splitting in two.
and mitosis is all about making identical copies, like
Right.
for growth or repair.
Precisely. The goal is genetically identical daughter cells, clones of the parent cell.
So what do the chromosomes look like when they're getting ready for this? You said they're duplicated in S phase.
Right. So before M phase actually starts, during interphase, GG$, S, G22, the DNA is relaxed, like long, thin threads called chromatin.
Hard to see, probably.
Very hard to see.
Mm.
But as mitosis begins, this chromatin coils up incredibly tightly. It condenses.
Ah, so they become visible.
Exactly. And because they've already duplicated, each visible chromosome actually consists of two identical halves called sister chromatids.
Okay.
And they're held together at a specific point called the centromere. Think of it like a pinched-in waist holding the two identical rods together.
Got it. So walk us through the stages of mitosis, the choreography you called it.
Okay, first is prophase. Chromosomes condense and become visible. The nuclear envelope, the membrane around the nucleus, breaks down, and this structure called the spindle apparatus starts to form. It's made of microtubules.
The cell's internal scaffolding.
Sort of, yeah. It's the machinery that will move the chromosomes. Then comes metaphase, meta for middle.
They line up.
Perfectly. All the duplicated chromosomes are moved to the exact center of the cell, forming the metaphase plate. It's like an equatorial line.
And the spindle fibers attach.
Right. Microtubules from the spindle attach to protein structures on the centromeres called kinetochores. These are like the handles the spindle grabs onto.
Okay, lined up, attached. What's next?
Action, anaphase. The proteins holding the sister chromatids together at the centromere break down.
They split apart.
They split. This separation is called disjunction. The sister chromatids, which are now considered individual chromosomes, get pulled rapidly towards opposite ends or poles of the cell.
So each pole gets a complete set.
A complete identical set. Then telophase. It's basically prophase in reverse. The chromosomes arrive at the poles, they start to decondense, relax back into chromatin.
The new nuclear envelopes form around the two sets.
Exactly. You end up with two distinct nuclei in one cell. The final step is cytokinesis.
Splitting the cytoplasm.
Splitting the cell itself. In animal cells, the membrane pinches inward, creating a cleavage furrow. In plants, they build a new structure, a cell plate, down the middle, which becomes the new cell wall.
And the result?
Two daughter cells, genetically identical to each other and to the parent cell, perfect copies. Mitosis equals fidelity.
Okay, identical copies are great for making more of the same tissue, but not for evolution, not for mixing things up.
Right. For that, you need the other type of cell division, meiosis.
Which is all about generating difference, variation.
Exactly. Meiosis is fundamental to sexual reproduction. Think about it. If gametes were diploid, like somatic cells,
Then fertilization would double the chromosome number every generation. $2 becomes 4 cells, then 8 tellers.
it would be chaos, unmanageable. So meiosis is a reductional division. It takes a diploid cell, $2 in, and reduces its chromosome number by half, producing haploid cells.
How does it do that?
By doing one round of DNA replication, just like before mitosis, but then following it with 2 rounds of division. Meiosis II and Meiosis II.
2 divisions.
Okay.
And something special happens with the chromosomes, right? The pairs.
Crucially, yes. The homologous chromosomes need to find each other. Remember, these are the chromosome pairs one you inherited from your mother, one from your father. They carry genes for the same traits.
They pair up.
They pair up intimately in Meiosis I. This process is called synapsis. They lie side by side, forming a structure that actually contains 4 chromatids total, 2 from each homolog. We call this a bivalent or a tetrad.
And this is where the mixing happens.
This is where a major source of genetic variation occurs, crossing over.
Explain that.
While they're paired up so tightly, non-sister chromatids, meaning one from the maternal homolog and one from the paternal homolog physically exchange segments, they break and rejoin with each other.
So you're swapping bits of mom's chromosome with bits of dad's chromosome.
Precisely, creating new hybrid chromatids that are mosaics of the parental DNA. These points of exchange, you can sometimes see them later under the microscope as X-shaped structures called chiasmata.
Wow. Okay, so that happens early in meiosis I. What then?
Then, in metaphase I, it's the pairs of homologous chromosomes, these bivalents, potentially with chiasmata holding them together, that line up at the metaphase plate, not individual chromosomes like in mitosis.
The pairs line up.
And here's the key reduction step. Anaphase I, the homologous chromosomes separate from each other and move to opposite poles. The pairs disjoin.
But the sister chromatids.
They stay together. Each chromosome, moving to a pole, still consists of 2 sister chromatids joined at the centromere. This is the moment the cell goes from diploid, $2, to effectively haploid in terms of chromosome number.
Okay, so meiosis the cell separates the homologous pairs. What about meiosis II?
Meiosis II looks, mechanically, a lot like mitosis. The cells that enter meiosis II are already haploid for chromosome number, although each chromosome still has 2 chromatids.
So it's like mitosis on haploid cell.
Pretty much. In metaphase II, individual chromosomes line up. In anaphase II, the centromeres finally divide, and the sister chromatids separate and move to opposite poles.
Chromatid disjunction this time.
Right. The end result, after cytokinesis following telophase II,
4 cells.
4 haploid cells. And importantly, thanks to crossing over and another factor, they are generally genetically distinct from each other and from the original parent cell.
Okay, you mentioned two sources of variation. Crossing over shuffles genes on the same chromosome. What's the other big one?
The other huge source is the random orientation of homologous pairs back in metaphase I.
How they line up.
Exactly. For each pair of homologous chromosomes, it's totally random whether the maternal or paternal chromosome lines up facing one pole or the other. It's independent for every pair.
So for humans with 23 pairs.
The number of possible combinations of just which chromosomes end up in a gamete is $2.2 to the power of $203.62.
Which is huge.
Over 8 million possible combinations just from random assortment before you even add in the near infinite variation from crossing over within each chromosome.
That really puts Dolly the clone into perspective.
Yeah.
Bypassing all that natural variation generation.
It really does. It highlights how central this managed randomness is to sexual reproduction.
Maybe we can make this more concrete by looking at some of those model organisms you mentioned. How does this 9 and 2I stuff play out in real life cycles?
Good idea. Let's take baker's yeast, Saccharomyces cerevisiae, super flexible.
How so?
It can just reproduce asexually making clones by budding off daughter cells. That's pure mitosis.
Okay.
But it can also do sexual reproduction. 2 haploid yeast cells of different mating types can fuse together.
Forming a diploid zygote.
Right. And that diploid cell can then undergo meiosis, producing 4 haploid cells called ascospores, usually held together in a little sac called an ascus. So it cycles neatly between Nina $2.
Okay, yeast is relatively simple. What about a plant? Like Arabidopsis, the little mustard plant scientists love.
Plants get more complex. The main plant you see, the leafy green thing, that's the diploid sporophyte, $2.
Right.
But it produces spores through meiosis, and these grow into tiny multicellular haploid structures called gametophytes. One type produces eggs, the other produces sperm.
So there's a whole haploid generation, even if it's small.
Exactly. And then in flowering plants like Arabidopsis, you get double fertilization. It's pretty cool.
Double. What happens?
Pollen delivers 2 sperm nuclei to the ovule. One sperm nucleus fuses with the egg cell that makes the 2-monozygote, which grows into the embryo in the seed.
Okay, standard fertilization. What's the second one do?
The second sperm nucleus fuses with another cell in the ovule, which is actually already diploid itself.
Whoa. So 9 sperm plus $2 cell.
Equals a $300 triploid cell. This develops into the endosperm.
And what's that?
That's the nutritive tissue in the seed. The food supply for the developing embryo, often triploid.
Triploid. Okay, that's different. Finally, what about us? Mammals. Like the lab mouse, musculus.
Yeah, mouse gamete formation, gametogenesis, is a good model for humans. And here we see a big difference between males and females. It's about resource allocation.
How so? In females.
In females, oogenesis, making eggs. Meiosis is highly asymmetrical. You start with one diploid cell, but the cytoplasmic divisions are unequal. Meaning one cell gets almost all the cytoplasm and nutrients at each division. The result is just one large, mature egg cell, ovum, packed with resources.
And the others.
And 2 or 3 tiny little cells called polar bodies, which basically just contain the leftover chromosomes and then degenerate. All the investment goes into one viable egg.
Makes sense. And in males, spermatogenesis.
Totally different strategy. It's symmetrical. One starting diploid cell undergoes meiosis, and the divisions are equal.
There you go.
4 functional sperm cells, all roughly the same size, streamlined for motility. It's about quantity and speed and not packing resources into one cell.
Different strategies for different roles. Okay, so let's try to wrap this up. What's the big picture takeaway?
I think the core contrast to keep in mind is this. Mitosis is about conservation, fidelity, making identical $2 somatic cells for growth, for repair.
Keeping things the same.
Right. Meiosis, on the other hand, is about change. It's about reduction, getting from $2 down to $0, and it's fundamentally about generating genetic variation through crossing over and random assortment, creating unique haploid gametes.
Preparing for the next generation with new combinations.
Exactly. That variation is the raw material for evolution.
Which brings us right back to where we started with Dolly.
Mm-hmm.
If, as you said, the whole point of sexual reproduction, of meiosis, is to generate all this incredible variation, that 2 plus crossing over,
Yeah.
then what's the ultimate biological trade-off? Or maybe what's the risk involved when an organism like Dolly just sidesteps that whole process, when it reverts back to making exact copies through cloning, essentially using a mitotic-like process at the organism level?
That's a fantastic question to ponder, that tension between the need for fidelity and the drive for variation.
It seems pretty central to genetics, doesn't it? Something
Yeah.
for you, the listener, to really think about as you connect these cellular mechanics to the bigger picture of life.